Hi, everyone, and welcome to the MLOps podcast. I'm your host, Dean, and today I have with me Ljubomir Buturovic. Has over 20 years of experience working on the intersection of algorithms, machine learning, and bioinformatics. He's currently the VP of Machine Learning at Inflammatix Bio, a company that is using ML to diagnose infections in blood tests, and he's taught applications of machine learning in life sciences at SF University. Ljubomir, thank you so much for taking the time today.
My pleasure.
So tell us a bit about the work that you're doing at Inflammatix. What does production in machine learning look like?
So first, some elementary background. First key product is called Triverity, which is a test for diagnosis and prognosis of infections in emergency room. And it consists of a device which we built and are currently preparing for clearance and launch, and algorithms which run on this device and which issue test reports to users. So the way it works, you take a blood sample. It's a venous blood sample from a patient, and you put the test tube with the blood inside a cartridge, and then a cartridge goes into the instrument. The instrument is called Myrna, and within half an hour, you get a test report on the screen of the instrument, and the test report tells you when the patient has an infection to begin with. If he or she does have an infection, what kind is it, whether it's viral or bacterial, and then also tell you what's the severity of the illness. And severity in our context means specific types of, the likelihood of receiving specific types of medical care, critical care, within 7 days of the emergency room visit. So that's the background. For legal reasons, I have to make clear that this product is not ready for sale or marketing because it has to go through FDA clearance, which we think will be imminent. But as of this moment, that's the step we still have to undertake. And following the clearance, it will be launched first in the United States, where we are based, and then maybe in other countries gradually. Making a transition to machine learning and ML ops, I mentioned an algorithm inside this instrument. That's what I'm working on. My team is working on. So our job, my department, is to deliver a software which runs on the instrument and computes the test report. So the software consists of two machine learning classifiers. One is diagnostic, the other is prognostic. Diagnostic means type of disease, and prognostic means severity of disease. The difference is that diagnostic classifiers, they predict, if you will, something that has already happened, meaning the person already has a disease. We wish to guess what type of disease it is. Prognostic means predicting something that will happen in the future. In this case, whether this patient will need critical care, which is very important in emergency departments. So the input features for these two classifiers are what's called gene expression levels or abundance. For your audience who is familiar with biology, these are molecular features. We use 29 specific genes that we measure, that the instrument measures. They're all numeric numbers, they're not categorical variables. And each value corresponds to basically the amount of gene product in the cells of this patient, in the blood cells specifically. This expression, as it's called, as I said, consists of 29 numbers, and that represents a file for each patient. And basically the product takes this, or our machine learning takes this profile which is a vector of numbers. There are also some normalization values added to this, but the biologically informative content is 29 numbers processed through machine learning classifiers, and they compute the probability of certain type of disease. For diagnostic, there are 3 classes: bacterial, viral, or non-infected. And for prognosis, there are 2 classes. It's severe illness or not. So you think about it that way, it becomes a classical machine learning problem, right? For you take as input a numeric vector of values, and you produce as output a probability of disease or probability of outcome. So once you translate that into this domain, then it should become familiar to all your listeners, even those who don't have exposure to medical type of problems. The big trick in this whole scheme is getting the instrument built which measures these 29 values accurately. And that's what our engineering department has been working on for years now. But now it's ready for clinical studies, soon hopefully ready for clinic. So that's the high level of ML that Inflammatix works on.
Right. Thank you for sharing that. On the points that you made about sort of the unique characteristics of the problem that you're solving, the measurements obviously happen within the device that you're building, but maybe counterintuitively to some, what you're also saying is that the actual inference and sort of deployment of the model is also on that edge device, right? Like the device is not sending information to the cloud, and that's where the predictions are happening.
That's exactly right. The predictions are happening on the device itself, and then they're just shown on the display. There is also connection to electronic health records systems, which is optional. So the test report can be deposited there, but it's not required. It can be operated completely standalone.
Okay, I see. And then in many cases, like we've seen in the past, when deployment happens on edge device, there are a lot of restrictions on the types of model that can be supported, and even on the process of getting a model sort of promoted to production. Can you share a bit about how the deployment process looks like for you? So assuming the data scientists in your team have built a model that they're happy with, what do they need to do to get it really on the devices or into the customer's hands?
So the process of this, I guess, relatively straightforward in the sense that we produce the software for machine learning, which is packaged as a Docker image. It is delivered to our engineering department, which packages this image as part of the whole system. The whole system runs internally embedded version of Windows operating system. Then that whole system, together with the instrument and embedded software, is shipped to the clinical sites, which used it for our clinical studies. And in the future, the same process will be used to build and ship instruments which are used in hospitals for actual clinical care. So, so the real challenge was getting to the point of having a system which can be deployed on an instrument. That's what it took significant effort and time to get there. But
you mean building the initial Docker container that would work with the system, or something else?
Yes, that and building the instruments itself.
Yeah.
So for the part of the challenge was that the instrument and the software, the machine learning, were built in parallel. So while we were developing our classifiers, we did not actually have an instrument which measured these gene expression values that we could use for training. So instead, we had to use other instruments which are commercially available to measure the input feature values while our instrument was being developed. And this was going on for years. And so we were essentially training our models on a surrogate of the Myrna instrument while the instrument itself was being built.
And the reason you could do that is you basically took multiple instruments that covered together the entire measurement regime of the instrument that was being built, and then used that data to train the model, or was there something like, let's say, easier than that?
So the problem is not combining together 29 measurements. There are instruments that can do it. The problem is that they are too slow and too expensive. What's the problem? So, for example, there is state-of-the-art technology which can measure these gene expression values satisfactorily, but it takes, let's say, 20 hours, 24 hours. It's also very expensive instrument itself, so that's unusable in emergency department.
Nice.
But we used it as again, as I said, as a surrogate for our instrument. There are other similar machines like sequencers, which can also produce high quality data, but it's also impracticable for emergency room. So that's the key restriction is basically turnaround time and cost.
That's interesting. 2 points that I like that you brought up. One is that you're basically saying that it's not that the problem in all its forms is previously unsolvable, but that the way we solve this problem in other cases is just not appropriate for the specific use case of emergency care. And that's basically the sort of main advantage. And the other thing that you're suggesting, which is maybe a good idea, not just for the problem that you were tackling, is that in many cases, the data to solve a problem is already out there. It's just much less convenient to collect it. So you might gain sort of an alpha in solving machine learning-based problems by collecting the data in a less convenient way or more expensive way, training the model while you're building out the more maybe cheap or faster data collection methods. I think that might apply to other like sensor-based ML problems that maybe listeners are.
Yeah, I think that's fairly accurate way to put it.
Amazing. And you take these models, which, if I understand correctly, are, as you said, more classical ML models, and you train them on this data. How do you build evaluations for these? Is this also like classical evaluation, like maybe simpler, I don't know, linear regression problems? Or it sounded like from what you said earlier that there might be some complexity, especially around the prognosis you said, right? The like severity detection, because maybe if you give the prediction early on, then you only know if you were correct days later or something like that. So I'm curious how you evaluate your models.
So it has to do with the separation or partition of data into training and validation, and tests, which I'm sure is super familiar to most of your listeners. I'll elaborate in a moment. Let me just mention one point you mentioned, which is the survival, which is the prognostic model, which is predicting the event. Again, in our case, we are predicting for critical care for the patient. So in theory, that's a survival problem, as I think you suggested, because you could say, okay, this person will need critical care in 3 days, and this other person will need critical care in 5 days, or maybe longer time periods, and this person will not need, critical care, which is obviously favorable, preferred outcome. So in theory, that is a time-to-event problem, but in practice, because there is only 7-day period that is but for our application, and why 7 days? That's a separate topic. It's a clinical question, but that's what we settled on. So in this case, there is now what's called censoring. Censoring is when patients drop out of study before meeting a pre-specified timeline. But if you have a timer of just 7 days, very few people drop out. There are some, but majority are still available for endpoint evaluation after 7 days. So at the end, it becomes a classical binary classification problem. Either there was a critical care or there wasn't. So in the end, we end up with, again, for both diagnosis and prognosis, with classifiers. One is 3-class, the other is binary. So now back to validation and evaluation of our classifiers. First, we evaluated a large selection of different types of classifiers, because for us what matters is obviously helping patients. So whatever works best is what we chose. And we empirically found that for these types of problem, the linear models were inferior to anything else and had some advantages. One of them being linear means being less likely to overfit in the actual clinical use. So that's where we landed, not because that was a priori bias towards that, but because that's what our analysis showed. So we split the data into the development data into training and validation. So just to be clear, in our context, training and validation are both development data. So both of these data sets were available to us, to the machine learning department, for the classifier development. Once the development is over and we get to the deployment point, that's the point we were discussing earlier, where we actually put the classifiers onto the device. At that point, they are locked and no longer changing. And subsequently, they went into clinical studies to evaluate effectiveness. In these studies, the data is newly collected. That's called prospective studies in medical applications, meaning they were not available at the time that we were building the product. They were newly recruited patients, and that's called a test set. And that's the final evaluation of performance of the system, which is independent of any prior work that we did. And the results that we obtain on this set of patients, on the test set, they're used as a basis for submission of the product to regulatory authorities. Again, we are initially working with the Food and Drug Administration in the United States and subsequently in other jurisdictions. That set result serves as the basis for the regulatory submission and clearance, and then subsequently marketing, because then you say to the physicians, okay, this is the performance that our system gets in terms of diagnosis and prognosis on independent adult patients.
So,
just a couple of more comments on the training and validation, which is the development data. The split there involves something called platform transfer and also data heterogeneity. The infectious diseases are extremely heterogeneous by nature, which means that the gene expression profiles, the input features, can vary between hospitals, it can vary between races, between different countries, between maybe social economic factors. So we had to split the data into training and validation such that we minimize the bias resulting from this distribution shift. The other factor, which is similar, related to this, but is more technical, not biological, is, as I mentioned, during the development, we did not have the instrument that we use in clinic. It was not available. So we had to ensure that whatever we built will work well on that final instrument. So to address that challenge, we used several different instrument types during the development, and I mentioned some of them earlier, and then split the training and validation data such that we use different instruments in these data subsets. And that way we were able to tune our classifiers such that the transitioning from one instrument to another had least impact. We also used relatively advanced normalization technique to transfer the gene data from one platform to another. In all these methods, we achieved best, which works in different hospital settings, in different gene expression instruments, in different subgroups of patients.
Nice.
So that was kind of the driving factor behind splitting the data in training and validation.
Mm-hmm. And did you sort of, assuming you sort of, there's this interesting factor here, which is that in most non-regulated use cases, you have a test set aside as you described, but then if you try to measure your model on it and you realize it doesn't work well, then you sort of go back to the drawing board, build a better one, and then test it again, maybe with new data. I'm curious how that process would work in your case. Like, obviously, I'm rooting for you that it works perfectly on the first go, but even later on, let's say, even if there's an initial approval, you want to improve the model for V2. How does the error analysis look like in cases where the test set is the model is locked in at the test set level, and maybe you're not even allowed to do error analysis on the test set or something like that?
So these are two very different scenarios that you described. First, the scenario is you are developing product, you finish development, you lock down your system, and then you launch your clinical study. It's called pivotal study for a good reason. Then you observe and analyze results on that study. If those results are negative, that has a major negative impact because you are not allowed to go back, tweak the classifier, and then run the same test data again. You need to launch another pivotal study. So what you can do, you can use that test data, put it in development, tune your classifiers, and then launch another pivotal study to evaluate that newly retuned classifier. But that has, as I said, pretty major implications for business. That's, in soccer, I think that's called sudden death, when they play until 90 minutes, until one team scores a goal, and then it's over. So if you miss this pivotal study, it's a significant trouble.
Okay.
You can go back depending on how much support you have from your investors, basically, but it's a big problem. The second scenario is, let's say your pivotal study looks good and you get a permission or clearance to market the product and you start using it in clinical care, and then you run further analysis and find out that you can improve the product. Then you can, the rules are less rigid. You might be able to reuse the data that was originally used for initial test set, depending on how much you looked at the data post-clearance. The rules aren't 100% rigid. Also, they are actually changing to some degree in the recent years in the United States. There is a process. So in the past, once you have a change to classifiers, you have to resubmit the application to FDA and get another clearance for the upgraded instrument. It can be shorter because you have already some basis to claim that this type of device works, but it's nevertheless pretty major undertaking to do it again. And so it can take years. But now the FDA introduced a facilitated process which you can describe in advance in your original application. Here's what I'm going to do to improve my classifiers in the future. And if they say, okay, that's good, I think that process looks reasonable and it's rigorous and you already have credibility. If they give you permission to use that, I wouldn't call it a shortcut, but simplified process, then you can do it faster without explicit submission again.
So you're referring to the predefined change control plan where you show your ML practices sort of as part of the initial approval.
That's it. Yeah. And that's going to be part of our submission.
Yeah, so I think that's one of the very positive things that are happening in the regulatory side with ML. There's a lot of talk about the sort of maybe negative or slowing down effect of some regulations, but this is a counterexample where earlier as you mentioned, you would have to go through a standard approval process every time, and now I think this is mainly for AI or ML in medical devices use cases. You can actually, it does require that you meet certain standards in how you manage the ML development process, but I think those are standards that are reasonable and healthy for every serious ML organization, like versioning your data and showing like how your models are tracked and how you compare different versions of your experiments, and that gives you much faster time to market in the second iteration and above, right? So I think that's
a great summary of the PCCP process. 100%, it's a step forward.
Amazing. So what, given that we just mentioned that you need to sort of work in an organized way and manage the efforts that you have, what type of tools do you use to manage your machine learning projects?
So I guess for code revisions, we use what everybody uses these days, Git, and we specifically use a Bitbucket for our remote repository, which is also very well established. I think for managing the data sets and machine learning models, we have maybe a somewhat unorthodox approach. We are very enthusiastic users of an app called Monday.com, which is used for tracking projects and I guess lists of items, you could say, and other organizing purposes. We found it extremely convenient to use for our data sets and classifiers. Part of the reason is that they are not that big. As you've heard, the input features to our model are 29 biological features, and the sample sizes are in the thousands. Managing different versions of these data sets in terms of storage, it's not a major issue. We just found Monday.com to be extremely convenient for this purpose. The other and similar argument holds for our machine learning models. We use the same platform to keep track, and we have some in-house built automation tools to interact with the .com. So it's been empirically frictionless for us.
Nice. Monday's offices are, let's say we can see their logo from our offices. It's I think 500 meters from us. So I don't know if we have any audience from the data science team there, but I wonder if they're using their own tool for data science or not. But I like that. That's creative, and if it works, then it works, right? Like that's the important part of a tool.
So far, as I said, it's frictions are minimal in that area. So that's where we are for the moment. Always open to improving productivity and flexibility. But for the moment, that's not a major area of focus.
So what is the biggest challenge that you're facing in the ML side of things? And how are you trying to solve it, or are you solving it right now?
I basically mentioned it. The biggest challenge is various types of distribution shifts in the both from biological and technical side. As maybe a reference point, I used to work for 10 years plus in cancer, and usually we think of cancer as very heterogeneous disease, right? Because there are obviously different types of cancer.
Mm-hmm.
But compared with infectious disease, cancer is a picture of uniformity. It's amazing how much variance there is in infectious diseases. And that's your challenge, which I think doesn't have a generic solution, at least to our knowledge, in machine learning. So we try to keep track of latest innovations in that area. But so far, the most effective tool to deal with this has been a collection of high-quality data. That's really what's played a major role. Well, one thing, actually, there's one more thing I should mention. So our development data is a combination of private data, which we collected from patients using clinical studies, and public data, which you can download from internet, from various databases like ArrayExpress and Geo. Those data are very valuable because they are public and they span a very diverse populations of patients in different countries and very different conditions. We spend a lot of time working on normalizing these data sets such that they could be utilized together because they are measured by the technologies and in different circumstances, and it reduces additional technical variation, which we spent significant time dealing with. But once we had a reasonably effective approach to handle this, we were able to significantly enlarge our training data sets. And
nice.
The other factor I should mention in terms of, which is also related to distribution shift, is the fact that for viral and bacterial infections, there is a seasonal variation because viral infections are higher frequency in the fall and winter, and they're less prevalent in summer months. So that's something you also have to take into account during development, and it also affects way that the system is used in clinical practice, because in winter times the physician will be more predisposed to conclude that the patient has viral infection, and vice versa for summer.
Man, that's interesting. I remember I'm probably going to butcher this because I'm not a biologist, but I remember I visited San Francisco just before COVID started, and I went to a talk by the Chan Zuckerberg Center organization, and it was about HIV. And there was a slide there that showed the genetic variants of different types of diseases and biological problems, and you see like the size, and then the I don't remember what the reference was. I think it was like influenza was like, you know, like this size, and then you have HIV, which is like huge, and that sort of explains why it's so hard to cure that, because it's so there's not really one type of HIV. It's like an infinite amount of variance there. So that's probably a very hard challenge that you need to deal with. I guess taking a step back, the three last maybe more higher level questions. Looking at the entire machine learning AI field, what are you currently most excited about?
That has to be really the advances in LLMs and related technologies in terms of, but I see it as primarily a tool for increasing productivity, which formally speaking, we feel that almost daily in our work. And I think that is a huge potential for those applications. Probably piggybacking to that question is what's the potential for that in clinical care and in medicine? There are a lot more cautious because I just explained some degree the process that has to happen before new technologies and tools like ours are introduced to clinical care. Similar rules should apply to LLMs and other innovations in AI in clinical care, and that's very challenging. It can be time-consuming, and it has to be done rigorously, and there's not even remotely a consensus among experts how to approach this in medicine. So we have to be, I think, very cautious there, because the first principle should be do no harm. So introduction of this, it cannot make patients' outcomes worse than they are.
Mm-hmm.
And also,
are you optimistic about the use of LLMs in sort of these clinical use cases, or do you think that we're a long way from that actually being practical?
Depends on the time frame, right? Whether I think it will be some years before we see widespread introduction. Whether it's considered optimistic or pessimistic, that's kind of judgment call. But I think it has to be rigorous. Also, if we have a minute, I want to mention that when discussing the prospects for either LLMs or AI or ML in medicine, we have to clarify what we mean by adoption, right? Adoption can mean different things to different people, but in medicine, there is something called standard of care, which is a set of essentially rules and guidelines which govern reasonable care for existing diseases. These are the procedures or diagnoses or interventions which physicians generally follow unless there is a very strong reason not to. Adoption basically means becoming standard of care. That's when some new technology in clinic is adopted truly. So I think the LLMs and our technology as well should be subject to the same metric. It is truly made it once it becomes standard of care in the particular disease area. And I think that's a yardstick by which adoption should be measured. And just for the record, there are very few ML or AI devices in clinical use which are standard of care. I think they could be counted on fingers on one hand. So that gives you an idea what's the challenge there.
Yeah, I think that's actually a nice yardstick, not just for the medical space. We usually talk in general, social media. There's a lot of talk about whether ML has found its real use cases in a lot of industries. And I think that we have people that you could argue are the early adopters in almost any field that are now trying to apply LLMs. But you could even say, for marketing, right? Like creating marketing copy, you can consider LLMs to have made it when it's the standard of how you create copy and not when you need to be like the early adopter or something like that. Now, I do think that in marketing specifically, we might be close to that. It's kind of hard to measure, but it's interesting to set like standards of care, like parameters for other industries and see where LLMs are in each one. You could maybe, in theory, you could maybe have like a progress bar of what percentage of people in an industry are using a certain technology. It doesn't have to be LLMs, and then define some threshold there, which is like, okay, this is now the standard, and that's interesting.
I'm glad to hear that it has more general ability outside of medicine.
Yeah, it would be interesting. I would be curious to see what the usage rates are of LLMs in different industries. So if you can maybe like to add some more spice to the conversation, if you could share something that you think is true, but that few people would agree with in the field of machine learning and AI.
So one thing I would nominate for this segment is what's called interpretability. And the basic point is in medicine, AI interpretability, in my opinion, is overrated. Just let me give some background on this, because I suspect a lot of people would argue that precisely in medicine, it should be high priority, but I don't think that's the case. First of all, this is based on empirical observations. I worked on this type of products for 20 years, and I spoke many times to many physicians about our tests in different areas, primarily cancer and infectious disease. Nobody ever asks about interpretability, not one single time. This cannot be just accidental. The one thing that all clinicians ask for, what is your validation data or test data? We called it earlier. What is the evidence that you have that this test is applicable for my patients? What is the evidence that it leads to better outcomes? What is the evidence that it can maybe reduce cost of care or duration of hospital stay, maybe insurance fees, things like that. But interpretability, it just never surfaces. If you have evidence that you can achieve all these desirable outcomes, then there's very limited interest in what happens under the hood. Granted, it is of high interest to machine learning experts and engineers, but for clinical and use cases is a lot less than is usually assumed. To give some additional points, I can imagine that the majority of ML applications in clinical care today are based on imaging. I think it's somewhere around 70%. I can see a case for interpretability in imaging applications. I guess you could point to a pathologist or other physician. This is the area of concern for these patients, which led me to conclude that they have disease X. So I imagine that is helpful. But outside of imaging, that's a lot harder to make that case.
It's interesting because in imaging, I think it's the hardest to build interpretability. So you're saying basically that where it's easy to build, it's not as interesting, and where it's interesting, it's not that easy to build. I like that. That's a good non-trivial take. That's great.
Again, based on my experience, that's where the things are.
Perfect. I think it would be like a fresh take for a lot of listeners, and I understand what you're saying. Like in hindsight, I think that when you talk to people, maybe the interpretability case, if I had to steelman it, would be that when things go right, no one cares about interpretability. But then if there's like a failure of the system, then you want to be able to say like it failed because of this thing. But I think from a like medicinal perspective, if you think about this and not like procedural, because if you go and you, I don't know, get a surgery done, an operation done on you, and there is like a mistake that leads to bad results, then there's going to be like an investigation into why that mistake happened. But if it's a regular medicine that you take, that has side effects, and those side effects have statistics, right? So you read the label and you make sure that the negative side effects are either very rare or you're not in the sensitive group with respect to those side effects. And then you sort of take a calculated risk. You don't ask in hindsight, like, why did it happen in this specific case? Because if you say that 1 in a million people will have more severe side effects, then you might be that 1 in a million, assuming that's what the data actually shows.
Yeah. Other parts, I mean, none of these systems are perfect or anywhere close to it. We have to live with some degree of risk, but that's what we have to today in classical medicine. So that's life.
Yep. And to finish up, if you have any recommendations to share with the audience, and it does not have to be data science related.
Yep, I have one question. I guess background is a lot of the news these days are kind of negative. If you want to feel a little more upbeat, check out the website called Our World in Data. I'm not sure if you're familiar, or maybe some of your listeners. It's the development economics portal at Oxford University. It's a first-class science. It's not some kind of low-grade resource. And they produce statistics and articles on different development characteristics globally and per individual countries. So there are hundreds and maybe thousands of graphs, and so it can be overwhelming. But if you want to feel better right away, check out the graphs or sections on infant mortality and the global poverty, and they both go way down for many, many decades in the past. So something I highly recommend for occasional reading.
Nice. I like that. Yeah, I'm familiar with Our World in Data. If any of the listeners are not, I really recommend it. We'll also add a link so that you can easily find it. I have 2 recommendations this time. One is I started reading a book apropos some interesting things that are happening online. There was the whole thing with OpenAI now that Ilya Sutskever, that had the falling out with Sam Altman, has now officially left the company. And in one of the tweets following his departure, Greg Brockman, who's another founder, and they're sort of like 2 of the main founders of the company, mentioned that he remembers when they read a book by Arthur C. Clarke called Profiles of the Future. So I immediately, when I read that, I added that to my reading list, and I'm now halfway through. I really like it. It's like Arthur C. Clarke, if the audience isn't familiar, is a science fiction writer. Most of his stories have like plots, and they're written well. Childhood's End is one specific one that I remember from earlier days, and I think it's a very good book. But this one is more doesn't really have a plot. He sort of takes different technological ideas, as he calls it, to their logical conclusion. So sort of futurism on many different axes. So every chapter, is dedicated to a different technology, transportation, teleportation, stuff like that. It's interesting. It was written a long time ago. It's interesting to see the things that he got wrong, at least for now. One example is he hypothesized that once we get a good grip on bioengineering, we're going to want to start to go back to riding animals, but we'd prefer to bioengineer a smaller elephant because they're not as dumb as horses. This is a quote. It's not my opinion. But like 1 page after that, he makes predictions on electric cars, which sound a lot like what we have today with companies like Tesla. So like miniaturizing motors into the wheels and things like that. So there's a lot of interesting reading there. Again, I'm halfway through, but I already recommend it. The other thing is, when you're listening to this, we'll probably launch this episode a bit earlier than we usually do. So I usually try to launch on the 15th of the month. But this month in June, we're going to be flying out to San Francisco and Seattle for the Data and AI Summit by Databricks. So that's the 10th to the 13th of June, followed by CVPR in Seattle. If you're listening and you want to meet up and you're in Seattle or San Francisco, then please reach out to me. I'll provide a link that you can just send me a message or connect with me on LinkedIn. And I'd love to meet you, any of the listeners, for coffee or a beer. And with that, Ljubomir, thank you so much for taking the time. It was a pleasure, as always, and I'm looking forward to next time.
Thank you for inviting me to your podcast. Very interesting conversation, hopefully for listeners too.
I believe it will be. So thank you, everyone, and I'll see you next time.
See ya. Bye.
Thank you for listening to the MLOps podcast. If you enjoyed this episode, please like the video or subscribe. If you have any feedback, leave a comment below. Thanks again for listening, and see you next time.